I think we should start now. It's 10:03. All right. So,
Yeah, that's good.
yeah. Bozo, Anne, and welcome to the sixth installment of our 2020 GLIER T-Rust virtual seminar series. My name is Smita, and I'm from Geophysics Lab at School of the Environment, University of Windsor.
And my name is Hector Esparra. I'm part of the Aquatic Disturbance Ecology Laboratory at Wayne State University as a PhD student.
And we are your host for today's seminar.
To begin, we would like to acknowledge that the University of Windsor and Wayne State University sits on the traditional territory of the Three Fire Confederacy of First Nations, Anishinaabeg, Peoria, Miamia, and Mississauga Nations.
We, as the Windsor and Wayne University communities, would like to move beyond acknowledgements, and we would like to not only give our respect to the traditional territory we are on, but also commit to learning and growing, starting with our partnerships and collaborations moving forward.
And to that end, we are excited to announce that we will open the seminar series with a traditional ecological knowledge panel in the near year featuring knowledge keepers and elders here in the Great Lakes Basin. Save the date for Friday, January 29 at 10 a.m.
Also, we are pleased to announce the final speaker for this fall season today, and in 2 weeks' time is brought to you from School of the Environment and University of Windsor. The date is Friday, December 18th. Dr. Paul Bowman from the Canadian Society of Exploration Geophysics, distinguished lecturer, who will present Good Geoscience in Dire Places, Searching for Water in Humanitarian Crisis.
As part of the Zoom ground rules, like everyone should already be on mute and have their video off, please confirm that. And if not, please mute and turn the video off now. Also, your chat facilitators today are Katrina and Jamie. Please send your questions to them, and they will pass them on to me or Smitha and the speaker. This talk will be recorded and posted online at a later date, and we would like to ask our technical team to initiate the recording now if it's not already recording. The format of the seminar goes as follows. Speaker will deliver a shorter seminar presentation, about a half an hour. Second, early career researchers are encouraged to stay for the second half, which is focused on career mentorship, Q&A style discussion with the speaker.
Thank you. And now I would like to introduce our invited speaker, Dr. Supriyo Das. He was awarded PhD in biochemistry by Stockholm University in 2007. After finishing PhD, Dr. Das continued his research as a postdoctoral researcher at NIOZ University of Glasgow, Örebro University, and Linnéa University. During his research, he received prestigious EAOG scholarship, British Council, and Swedish Research Council fellowships. He is presently working as an assistant professor at Presidency University in Kolkata, India. His research applies elemental, molecular, and stable isotope geochemistry to understand the interaction between continually changing climate, environment, and human activities. He was a member of IODP Expedition 354 and 38th Indian Scientific Expedition to Antarctica. Dr. Das will present a seminar entitled Alkane for UV Screening, Ask Antarctic Algae and Moss. Over to you, Dr. Das.
Thank you, Smita, and thank you, GLIER T-Rust, for giving me an opportunity. And good morning to my Canadian audience. Good evening to the audience in India. So throughout my research career, I actually worked with phytoplankton or primary producers in freshwater and marine systems. I looked there, one of the first few occurrences in geological record to the very recent blooms in the water. And what I found is that their productivity, their primary productivity in aquatic environment is primarily controlled either by the anthropogenic activities, that means like pollution, or the climate change, or sometimes both. Now, I don't really study the living cells or living organisms under microscope. I actually do the molecular and stabilized analysis of these samples to find the record so that these signatures, their signals they are keeping in them could be used later for paleoclimate and paleoenvironmental reconstructions. And that was one of the objectives, research objectives for me, or the motivation for me to go to Antarctica. And I was a member of 38 Indian expedition to Antarctica. I spent there a few months in summer, Antarctic summer, and I was stationed in the Bharati Research Station in Larchman Hills. Now, this is a very weird looking and very beautiful looking in other way station, quite, fascinating. And I was there from 9 December to 15 December 2019. When I was in Larchman Hills, it was very, very warm. So I'll talk about it later. Before I should introduce where Larchman Hills is. It is in the eastern Antarctica. And in this, I think I could minimize this. Yes. In the map on the right hand side, you see the Larchman Hills region, which is basically divided in Grafness Peninsula in the north, Brockness in the south, and there is Fisher Island in between. And at the right upper corner, we see the Elizakate Island. Now, these are the areas, primary areas, where I actually sampled and I studied during my stay. As I said, when I went to Antarctica, it was very fascinating, and I was, very surprised at the same time because throughout the time, and throughout my life, I never saw a barren place like this. There is no green, and you may know that there is no rainfall in Antarctica. And East Antarctica is the warmest and driest part. Uh, there is no ice in the sense like there is no ice sheet. Of course, like snow accumulates during winter and early spring to summer, most of the snow gets melted. So it is very warm when I landed in Antarctica, and it was so warm that we could work in shorts, but we are not allowed, of course. So it was 6 degree, around 6 degrees Celsius during daytime. If it is not windy, it is really hot. If it is windy, we can feel chiller. Chiller means we can feel very cold. So we are not allowed to go outside without our proper Antarctic gear. So in this two pictures, you can actually see small, ponds or like water accumulations that was due to the meltwater. Uh, Before winter comes, at least the left hand side pond or the zone will get completely dried. The right hand side is a murk water, which probably stay there somewhere, sometime, to some extent, completely frozen. But that is not very deep. Now, if we are talking about real lakes, there are plenty in Larchman Hills. There are over 120 lakes in Larchman Hills, which are quite deep. Probably, well, deep means not very deep, probably a few meters, 7-8 meters, not more than that. And these lakes are oligotrophic. You see there this clear water. Basically, if you can go in the middle of the lake, and if it is frozen or partly frozen where you can stand, you can see the bottom, which is often very scary for people who are not used to look into the lake bottom from the surface. Uh, so here we find algae mostly under the water, in the lake bottom or in the shallower parts, which is really moist. The moss beds, they are not very far from the lakes, but they are confined in the moist areas, mostly in the flow, in the meltwater flow channels, where they can get enough moisture and nutrient. Now, these are the basic plants or sort of things in Antarctica. You can find lichen and a little bit of fungi, and that's it. You won't see any plants, at present, and no vascular plants in main mainland Antarctica, continental Antarctica. So in our Larsemann Hills, there is nothing. There's no grain. Of course, like you can see grains, and that's how I can tell you. I mean, the moss beds in Antarctica, they often change their colors. They can be green, they can be red, they can be gray, or even black. And this color change is linked with the availability of water. Like if they have plenty of water, they're green, they're healthy. If the water gets less, like if it's getting drier a little bit, they change their pigment color. So what we know so far from from the study, not in Larsemann Hills, but elsewhere in eastern Antarctica, that this change in pigment are linked to the water stress. You know, uh, some researchers, they actually linked this color change to the UVB radiation. So these are the basic understanding I had before going to Antarctica, and I know that the color change has some geological or geochemical connection, because they are connected to pectin and ester compounds in the cell walls. So these are the compounds that might be of my interest. Later, I have found more interesting thing that I will tell you. Now, in Antarctica, I don't see a very clear and prominent change in color in the moss. I do found uh, green moss, and those are mostly in shades. Uh, and mostly the moribund moss. Those are very dry moss. These are very dry. I mean, if you can take a sample, like if I could show you a sample, I could demonstrate. So they are very dry. Sometimes they are a little wetter, and they may have some green or reddish spot, and they are very thick. They are like blanket on their surface, and they are mostly, I mean, found along the meltwater channel, which is dry right now, which was probably moist at some point of time, probably early spring. So this moribund moss indicates complete loss of pigments. They are completely dry, and they are dry mostly because of the water stress, and they have lichens all around. We have at least 3 species of lichens in Larsemann Hills. Here you can see 2 of them, and I studied those as well. But in this talk, I will not talk much about lichens. I will probably concentrate more on the moss and a little bit on the algal mat. Now, when we talk about extreme weather in Antarctica, I mean, we probably consider temperature. Of course, it is. Though Larsemann Hills is very warm most of the time, say maximum 6 degrees Celsius, but it can be very cold. The point is that it could be very windy. If it is windy and cold, it means it is extremely dry. And it is extremely sunny at some point, mostly during the summer. And Antarctica is infamous for the extensive ozone depletion and UV radiation. So we were not allowed to go outside without sunscreen. We always applied sunscreen on our skin. But plants like moss, they don't have this luxury. They cannot go to the supermarket and buy a very, protective sunscreen. So they had to survive with that. They have to uh, do something to survive under such a condition. The algal mat that lives underwater, they are a little bit fortunate, I should say. Although the lakes are oligotrophic, and we may consider that these oligotrophic lakes may not protect much from UVB radiation, but logically, if it is underwater and quite a little bit deeper, probably they receive little less of UVB radiation. So that is the basis of my research and talk here, where I expected my hypothesis is that algae living underwater receives less amount of UVB radiation, and moss, which lives on the ridge and exposed to sunlight, receives more UVB radiation. So that with this hypothesis, I see what I know so far about Antarctic moss and their UV screening capability. So far, whatever we studied about Antarctic moss and their UV screening, we found a clear link with the pigment. Even the bacteria, cyanobacteria, for example, they have the capability of producing UV screening compounds in their membrane. Elsewhere, in other part of the world where you have vascular plants, their epicuticular works as inalkanes, and those inalkanes they also used for UV screening. But in Antarctica, we know almost nothing about the inalkane synthesis by moss or algae, and the screening compounds that they use, we don't know their exact composition. We roughly know what they broadly know their chemical composition, but not exactly. And especially if alkanes have any role to play. Now, alkanes are hydrocarbons. They are, simple saturated hydrocarbon. The carbons are connected by covalent bond. And in Antarctic samples, I found in alkanes, those are normal alkanes in full. In alkanes from C17 to C30, that means carbon with 17 atoms and with 30 atoms. So those are the alkanes I found. It is quite a bit, and I can tell you that this sort of inalkane signature in algae or moss is not very common. This is very unique, but I'll not go much into this right now. The odd over even predominance and carbon preference index values are also quite high, and that is also extraordinary finding. When I say extraordinary, I actually compare my data, my finding with similar signatures elsewhere. I mean outside Antarctica. But this is not the most interesting part is something that I'm going to tell you right now. What we know about alkane in plants is like they have very distinct role against desiccation, and they actually, plants, vascular land plants, they actually produce short chain in alkanes to protect against desiccation and often against UVB radiation. Now, if the function of inalkanes are the same in mosses and algae, that we don't know. And basically, we even don't know how these organisms produce inalkanes. So there are a lot of, I don't know, stuff here. What I wanted to do, I actually analyzed algae from different depth with different color from underwater from lakes and mosses with different color and simply analyzed inalkanes in those samples. There are many observations. The most important, most interesting thing that I found is the ratio of short chain versus long chain. Long chain means C21 to C30 and short chain is C17 to C20. So when I analyze these two group of inalkanes, I found very interesting observation. Now, see the green mat, here the samples are from underwater. The green algal mat is at a deeper part of the lake, and the gray mat is at a shallower depth, like few centimeters. I could put my hand down and pick it up from the water. Although it is very, very, very cold, it is freezing cold, it's burnt cold. But I actually did that. But anyway, with decreasing water column depth from left to right, we see the ratio is decreasing. So very high ratio in green mat, probably because green mat, they don't need to produce inalkanes for protection, or they don't need much protection from UVB radiation. So they probably produce less of lower carbon number inalkanes. But the gray mat, they're very much exposed, they're very shallow depth, they're exposed to the sunlight throughout the day, 24 hours, and receiving too much of excessive UVB radiation. They may need to produce a little bit higher amount of shorter chain inalkanes. Now in moss, it is different because moss are exposed throughout the day, and they don't have any water column above them. some moss they can stay in this in the shade, but still like it is not 24 hours shade, so they are exposed at least most of the time during the day to UVB radiation. But their alkane production is not straightforward, because unlike algae, which is living underwater, they have water, plenty of water. Moss they don't have. So some mosses are uh, stressed, and I mean they don't have much water. They are dry, and some are little bit lucky, they still have water. So we can divide two groups. One is green and gray moss, and another one is moribund and dry moss. Now, moribund and dry moss, they are water stressed. They don't have much water. Their source of water is dried already. And what they do here, they actually produce longer chain inalkanes to survive during the drought condition, the water stress. That's a very common mechanism that they do. We know about it. The green and gray moss, they have water, so their stress is less, water stress is less, but they are exposed to sunlight. So that is their, the UV radiation is their primary stress here. And they probably produce higher amount of inalkanes, sorry, short chain inalkanes, and they have a lower uh, ratio. So with this, it is expected that this moss and algae, which is living underwater, has a very similar but not very close in alkane synthesis procedure. Now, green moss, when they produce higher amount of short chain in alkanes, they are still producing higher amount of, I mean, considerable amount of long chain in alkanes, because they knew they are going to get dry, or like their water source is limiting every day. And it is very windy in eastern Antarctica, and wind means that they are getting drier day and day. So with this, it is clear that when we stack samples which is underwater, has no water stress, and which is above water, has may or may not have water stress, they produce inalkanes for two different reasons. One is quite close to each other, but another one is not. The algae, which is living underwater, they produce short chain alkanes to protect themselves from UVB radiation. The moss, which are exposed, they produce short chain for UV screening and long chain to protect themselves from the water stress. Now, with this, I can conclude here that the alkane are produced in moss and algae. They are also produced in lichen, but I didn't tell about much because that will be a little bit different. The process, the mechanism that lichens follow to produce in alkanes. But what I could see that the contribution from lichens to moss is minimum here, because their volume is very less. So in algae, high amount of short chain inalkanes are mostly connected to the UVB radiation, whereas in moss, the UVB radiation and water stress both actually control the synthesis of inalkanes. Now, with this research, I must thank to my students who are working with me at Presidency University, and my collaborators from Presidency University, BSIP, and IISc Bangalore. Of course, the references I have a list I can share with you if any of you are interested. And here I end my formal talk, but I would like to show you a video, because in Antarctica, if I don't show the video, it doesn't end properly. So please stay with me. Well, that's all about my talk. I think I finished within time.
Yes, you did, and it was very nice. Thank you, Dr. Das, for an awesome presentation. Hector, you want to go ahead for some questions?
Yes, please.
You are muted, Hector.
Sorry. Yeah, some of the speakers like raise their hand. So yeah, let's see. Okay, so all right. Okay, so you want to start first, Smita?
Sure. So I have a couple of questions from the audience before we uh, s- uh, start looking for questions from early career researchers. So I have a question from Molly M. Does the Antarctic ozone hole affect the amount of UVB radiation that these monsters are receiving throughout the year?
Yeah, it is throughout the year, but it actually became very extensive during the springtime.
All right. Another question from Ryan Graham. It says, how does your research build on the effects of climate change in Antarctica?
One more time, please. How my research?
How does your research build on the effects of climate change in Antarctica?
My research on climate change in Antarctica,
Yeah.
and that, yeah. Well, see, climate is changing, and we know ice is melting everywhere. So Antarctica is not an exception, but what we don't know how it will affect us. I was reading a Nature's paper last week, and it says that even if we stop burning fossil fuel, adding carbon in the atmosphere, we cannot stop the climate change. We cannot stop the increase in temperature in next 50, 100 years. So we have to be prepared for what is going to happen, and how we prepare. We have to know every aspect. And what we know that, okay, it will become warmer, sea level will be higher, and people need to be moved. We don't know how these primary producers, they will react, they will respond to this climate change. And that's very much important, because even if we don't look at them or, consider them significant, they are higher in number than us on Earth. So we need to know how they're responding, like if they are going to affect our, agriculture, if they're going to spread some disease, or if they're going to kill other plants, or if they're completely getting extinct in near future. So all these things need to know, and we have to find places where we could do this research, because anywhere you go, there is anthropogenic influence, like people are polluting. So we need to know which part of the influence is from climate change, and which part of the influence is from the human activity, pollution, for example. Antarctica is one place where we don't have much pollution, which is pristine. So any change happening over there is, of course, from the climate impact. So that is the only reason, or the major reason, I could say, for me to go to Antarctica and study those changes, those signatures in algae and moss.
Thank you. So I guess there's no more questions from the chat per se.
Okay.
So yeah, let's move on to the Q&A. So yeah, a couple of questions that we have in mind for you is like, what type of advice would you give to early career researchers to have a successful career in your field of work?
Well, day by day, it is getting very challenging to
Yeah.
to make a
I mean,
living in academia.
I, I was watching the video. I was like, oh my God, it's so cold. But
Yeah, I
yeah,
don't like cold either.
nonetheless, it's interesting. I mean,
Yeah, I don't like cold either. People, I mean, I lived in Sweden for several years, and people knew me from Sweden. They said like, okay, Supriyo, you were the last person we could think of going to Antarctica because you left Sweden because you don't like cold. Yeah, I don't like cold. But anyway, when it comes about the making a career in science, uh, like there are twofold. One is that you do your study and you join industry,
mhm,
uh, especially for geology students or geochemists, students who are working in geochemistry, they have this option open. And you can choose to stay in academia. The difference, in my opinion, is that academia gives you a lot of freedom. Like I can go to Antarctica. If I don't like to go to Antarctica, I could go to, a desert, for example. So it's up to me. Nobody tells me like, Supriya, you go there next day. And it is my choice what I will pick up for my research. um, you know, and it is fun, like, and that much, but to reach here, you need to study a lot. Like you have to have a PhD, you have to do several postdoctoral research before you can actually end up in academic job that you actually want for. And you have to be very specific and focused, on publishing continuously. You have to publish throughout your. I mean, even if you are not doing any active field work or analysis in the lab, you have to look for something that you can publish. So that is the key. It is our currency. Publishing is our currency. We have to do that. And even right now, the world is under recession, but there are a lot of pockets of money. Like several societies, they actually pay a little bit to PhD students or MSc students. So you have to look for that. You have to apply. It is not that you apply and you get it. Less than 10% actually become successful. But with one and another application, you will find more opportunities, and you probably become more experienced. So I think I always ask, like, when you apply for opportunities, whatever it is, for visiting lab, for getting some small fund, for publishing a paper, and that is something is, I think, the very basic thing. If you want to survive in academia or in science, you have to look for opportunities and you have to try for it.
thank you.
Any more questions?
Smitha, you're mute.
Can you hear me now?
Yes.
Okay, so could you share your educational and career pathway and how it has led you to where you are now?
Well, I don't have a PowerPoint for this. So if you expected me to prepare one, I didn't do that. Sorry.
No, just give us some idea how you actually reached, how you got the opportunities, to finally get into this position, that you are able to do your research. And I mean, the research which you really like. So, what do you think, what were the advantages or the opportunities you got to reach to this point?
Well, a few things that I wanted to share. I mean, with my experience working in Europe, mostly in Europe, and I might not be very different in US as well, but I don't know about Canada, probably the same. So in academia, there are two kind of positions. One is teaching, and the one is research. The research positions are mostly contractual. So you have to prepare that you will teach when you do research in academia. If you think you will only do research, that is not good. And like, it is not good only not for position wise. It's also not good because when you teach, you face young mind, and they can ask you anything. You don't know what they can ask, and then it will prepare you for new questions. So it is important that you teach. So teach and research, that are the positions that you should look for. That's the first thing. And the second thing, when you you know when you select your postdoctoral research during the PhD, you don't have that. We don't have much option. We probably don't have much liberty either to divert much from the original project. But if you have the opportunity, do that, because that is how it will train you to survive in future. You can get a job, that is not a problem. For example, in US universities, those are training track, but they will expect you to earn enough, I mean, to fetch enough funding. If you don't do that, you may not have your contract, continued. So how to find a new problem? How to find, I mean, get more research money? That is the key to survive here. So for that, you have to train yourself, and yeah, I mean, that is sometime I think the how to call this, the employers, they also expect that you will earn fund, you will bring fund uh, for in your position. So I think, I mean, I don't need to tell where to get those advertisement. You know where to find job. That's not a big deal. I mean, everything is in our reach today. But how to crack it, that is the problem. And my way of solving is that, you become more competitive. You become uh, different in a sense, like you have to have the imagination to find out a new problem. Whenever I talk to my students, I say, like, see, the scientists are, they're like, they're philosophers in one way, and they also like dreamers. They can think of something that normal people don't think. Like whenever you invent something, discover something, you actually expect that something is there, or something you can make or build or something like that. So you have to be different from regular people. And in natural science, you have to be knowledge, you have to have the knowledge to be different, because science has advanced so far. You need to know many things. You need to know, biology, you need to know geology, you need to know physics, chemistry, math, even history or social science. You need to know many things. And for one person, if you are not genius, it's difficult to know everything. But you need to know a little bit of everything to find out new problem, and you can ask for, position or funding to get the answer to it. So I think I'm not very point to the question, but to the point means, like, where to find a job is, I don't think that is very important, because people know where to find a job, but how to, and how to apply, but how to get, that's the problem. And I think I sort of addressed that.
Yeah. We have some more questions from audience. I think we do have some time, I believe. So maybe I'll take one or two. So this one is from Madhurima. Why is odd carbon number alkanes preferred over even carbon number alkanes?
Okay, why this odd carbon number prefer when, okay, goes over even? So that is the way the alkanes are synthesized. So in terrestrial vascular plants, what mostly the alkanes are studied, they have their specific function to circulate or to control the water loss from their stomata. And here, the long chains, they actually give the, facilitate them to control the water loss from the leaf. And how it is done, like, if you, in a simple term, if you think that long chains are more robust than short chains, so you can think of this line. Another way of thinking is that there are certain genes that actually synthesis alkanes, and the alkanes are primarily in living cells. Alkanes are produced from fatty acids. So from fatty acid to alkane transformation, there are certain genes that become activated, and they work on certain conditions, like, for example, osmotic pressure or some other pressure. Mostly in land plants, osmotic pressure is the major controlling factor, and also in the moss, I guess. So that is how the trees still have land plants. They have odd carbon chains more than even. But with time, I have that feeling that it is not, it is not always true. At some point, it's true for land plants, but it is not true that algae or uh, phytoplanktons, they cannot have this odd or even predominance. They can have, depending on the pathway, they select to synthesize alkane. So I think I I answered your question. If you have more insight, you have to read into that, because specific species or plant type have their own way of producing alkanes.
All right. We have more questions from the audience. Okay, so these questions comes from Abhishek. Sorry if I mispronounced your name. He or she thank you a lot for the exciting talk, and would like to know whether any studies have been done on phytoplanktons, which form the base of the Antarctic food web and sustain the wealth and diversity of life for which Antarctica is renowned.
Any work on phytoplankton? Whether any work is done on phytoplankton that forms the Antarctic food web?
Yeah.
Yeah, there are a lot of work done. I mean, we know, but the point is that Antarctica is not a small place. So if you are working on western side, it's different than if you're working on eastern side. Not all places are are equally covered, and not everything is well known. So people are still working, but yes, of course, there are a lot of work has been done on phytoplanktons in Antarctica.
All right. So more questions from the audience. I don't think we have yet. So let's continue with the Q&As. So basically, GLIER and TIROS are very interdisciplinary programs that are focused on different aspects of environmental sustainability research, from blue skies discovery-based efforts to ones that are applied and relevant to decision making, practice or capacity building, among other things. So how do you try to incorporate different disciplines and local communities into your work?
See, for this, we often like to stay in our comfort zone. Like if I am a geologist, for example, I will only work with rocks. I will not look into anything else. So first thing that you have to address your comfort zone. You have to break out. You have to come out from it. And then when we identify a problem, we have to find a solution. Okay, some people approach is like towards problem, some people's approach towards solution. So if your approach is to solution, then you know what is lacking in your in expertise, what you need to know that you cannot tell from your experience or from your collaborators. In my personal experience, I worked with many people, from science people like, biologists, geologists, botanists, from people who works in history, sociology. Um, so, whenever I find I need something that I don't know, or it's better that an expert comes into the research, I go and find a colleague who can tell me more. I mean, of course, like we can study, but like if somebody is working in history throughout his life, is a better person to tell me about Indian history or world history than I'm studying history books right now. So that is how you have to find out your collaborators. And one thing that I think is like today, what we consider as blue sky research may be applied tomorrow. There are a lot of examples. I don't want you guys know more than I do. So we may not know what is the exact application, but there are applications. Like when we studied phytoplankton or algae back in 60s, we probably didn't expect that one day we will study the same thing for producing biofuel. Now we study, I mean, whenever I study the research papers on Antarctic algae, I found more papers on algae that are used for biofuel. So I don't think in 1960s or when we started studying organic geochemistry, we had this idea in our mind. So it was blue sky at that time, but now it is more applied. It's industrial research, and you get a lot of funding if you are expert in doing that. I think I roughly, answered your question. I'm not sure.
Yeah, I think, yeah, I got it. I have another question, and I think most probably this is going to be the last one, looking at the time. So this one is from Mercedes. Are there plans for a long-term study to investigate whether the carbon chain ratios exhibit ongoing change? For example, in response to climate change?
Are there one more time?
I'll repeat. Yeah. So are there plans for a long-term study to investigate whether the carbon chain ratios exhibit ongoing change?
Yeah, of course, like there need to be ongoing study, like somebody need to sample it properly. Like we went there, we didn't know what to expect. Now we have a little bit of sample from each kind of, not kind, I should say, it's color of mosses and something like that. But it need to be done probably at least few more years to see if it is a, just isolated finding, or it is like a trend, or it is there. So we need to study longer, and we need to study more into the synthesis of these carbon molecules, alkanes, I could say, in those plants in Antarctica. And if you do a literature survey, you will find there very little information from living plants from Antarctica. They study soil. For us, it is very common to study soil or sediment, find out the alkanes and link them to past changes. That is the very regular procedure. We hardly look into the living stuff and find out exactly the source we are finding in soil or sediment is the same in the living organism. So that link is not there in Antarctica. That need to be established, and for that we need to study longer. We need to do repeated studies from a different part of Antarctica probably, and more samples. That's true.
Yeah, thank you, Dr. Das. And from all of us at SOE, School of the Environment, GLIER, and Trust programs, we would like to extend our thanks to you and to all who attended. And with this, I would like to share our next speaker, which is on December 18th, Friday, Dr. Bowman. Thank you, everyone.
Thank you. Bye.
Bye.
Yeah, have a good day.
Have a good day. Bye.